Welcome to Homework Lib.
Hello students, the question asked is explain how cell types specific selector genes such as MyoD control cell differentiation. And we have to discuss in detail the molecular mechanisms by which such genes regulate activation of other genes. As we know that the cell differentiation, it is an important process in the development of multicellular organisms as it involves the formation of specialized cell types which have specific functions. And one of the key factors that control cell differentiation is the expression of cell type specific selector genes, such as MyoD. And these genes play a significant role in determining the fate of a cell by regulating activation of other genes which are involved in the developmental process of a particular cell type. MyoD, which is also known as myogenic differentiation, one is a transcription factor that plays a critical role in the differentiation of muscle cells. And this MyoD belongs to a family of proteins called myogenic regulatory factors, and which also include MYF5, myogenin, and MRF4, myogenic regulatory factor 4. And these proteins are essential for the development of skeletal muscles as they control the expression of genes required for muscle cell differentiation and function. The mechanism by which MyoD controls cell differentiation can be divided into three main parts. The first one is activation of MyoD expression. The expression of MyoD is regulated by various signaling pathways and transcription factors that respond to developmental signals and environmental signals. For example, during embryonic development, the expression of MyoD is induced by signaling molecules such as WNT and sonic hedgehog, which are secreted by neighboring cells. And these signaling molecules activate specific transcription factors such as PAX3 and PAX7, which in turn bind to the regions regulatory regions of the gene MyoD and promote its transcription. Another one is the binding of MyoD to target genes. So once this MyoD is expressed, MyoD protein is expressed, It acts as a transcription factor, which means that it will activate the expression of other genes. So it binds to specific DNA sequences of other genes. Those specific sequences are known as E-boxes and which are present in the regulatory regions of the target genes for which this MyoD is responsible for the activation of those genes. So MyoD forms a heterodimer with another protein called E2A, and this complex recognizes and binds to E-box sequence, and the binding of MyoD to a target gene is a crucial step in the regulation of gene expression. As the binding of heterodimer of this MyoD and E2A recruits additional transcription factors and chromatin remodeling proteins that facilitate the activation or expression of the target gene. Third one is the regulation of the target gene expression. After the MyoD has bound to the target gene, it regulates the expression of, by modulating the activity of transcriptional machinery. This can involve the recruitment of coactivators such as histone acetyltransferases, which modify the histones and promote a more open chromatin structure, allowing for increased transcription. Conversely, MyoD can also recruit co-repressors such as histone deacetylases, and which remove the acetyl group from histones and hence deactivates or promoting the condensate chromatin and leading to transcriptional repression. I hope you found the video useful. Thank you.